So this section is going to look at plant tissues, and your learning outcomes are to name the 3 types of ground tissue and their functions, and then your second one is to distinguish between xylem and phloem structure and function. So here there's going to be 3 main types of tissues, and I found a diagram to kind of help illustrate. This would represent a young seedling, and there would be your meristems there. You'd have another meristem down here. From those cells, as they differentiate, which was talked about in the prior section, they're going to differentiate and go down paths that would give rise to cells of certain types of tissues. The 3 main types of tissues that will be covered are dermal, which are on the outside or the external surface of the plant, and they provide protection. And then we have ground, and this will form several different types of internal tissues. Some of them are going to be involved in photosynthesis or storage or provide structural support overall. And then we have the vascular tissue, which is going to be conducting water, minerals, and nutrients. And so you can see this highlighted here. They section through a leaf. They also section through a stem and through a root, and then they show the cross section just in diagram form. And the blue structures that are within any of these represent the vascular tissue, which is for conducting water and things dissolved within it. The green is supposed to represent the ground tissue, and then the dermal tissue is in yellow. And you see that that's going to be seen in all structures throughout the entire plant. So we're going to focus first on dermal tissue, and I went ahead and found a section through a leaf. So we took a little and cut a little part out of the leaf there, and then looked inside the leaf. So inside the leaf you would have cells that do photosynthesis, and they call that mesophyll, and we'll look at the structure there of that later. But looking at dermal tissue, you can see that the top part, which is up here, and the bottom part of the structure is going to be lined with a layer of cells. That layer of cells is called the epidermis, which is made of dermal tissue. There's going to be special structures like trichomes and guard cells that form the stoma. Those are special dermal cells that have differentiated for specific jobs. This layer of dermal tissue is going to be 1 cell layer thick. Its function is for protection. It's often going to be covered with a waterproofing material. On the case of a leaf, it would be a waxy material called cutin. And they call this layer, it's not living, but it's produced by the cells, would be the cuticle. And again, you're going to have many special cell types, including those guard cells, trichomes, and down in the roots, in a very where new root growth occurs, you're going to have root hairs growing there. So taking a look at the guard cells that we saw underneath the leaf there, it's a pair of cells that are specialized to change shape. The notes here refers to them as sausage-shaped cells. So you can see in the first image here, which is a scanning electron micrograph, this would be one of the cells, and then the other one. And so as the pressure changes in the cell, the plant can regulate pressure by moving potassium around, for example. Wherever electrolytes go, the water would follow. And so if we moved particles like, say, potassium into the cell, that would draw water in, and that would increase water pressure, and that causes those cells to bow outward, and that gives us an opening here, which is called a stoma. And that stoma is used for the plant leaf to exchange the atmospheric gases. The plant's going to be producing more oxygen than it does CO2. It does produce CO2 because they do cellular respiration with mitochondria. But during photosynthesis, they produce a lot of oxygen. They got to get rid of it. So that comes out from those openings, say on the bottom of the leaf. And then they got to take in carbon dioxide, as you know, is required for photosynthesis. Another thing that exits out here is the water that goes from the roots and the vascular tissue all the way up the shoots and to those leaves. So water vapor also comes out of there. So you can see just different views. Here's another image, not as magnified, but still a scanning micrograph. You can see the guard cells there and the guard cells there. And in fact, the translation for stoma means mouth. So it's like a tiny little mouths there. Stomata is plural, by the way. And so the pattern of the production of these stoma cells is is done through done during the development of the dermal tissue as to whether or not these cells are going to be come guard cells or not. We don't just want a whole bunch of guard cells all over the place. There has to be some control over that. So we can see here in this image what happens when things go wrong. Here, when you have literally too many mouths, which is stoma, there it is, the TMM mutant. Here, this one shows the surface of the bottom of the leaf where too many stoma because we lost control because of the mutation in a gene involved in control of the pattern in which these guard cells are placed there. Then you have your basic epidermal cell there, which is going to be covered with waxy coating. Now, you might recall that when the plant is water stressed, they'll reduce the water pressure in the guard cells, and that closes the stoma, which helps conserve water, which would be maybe more important than exchanging atmosphere gas. Then we have trichomes, and trichomes, they may be single-celled or multicellular structures on the surface of the epidermis, and so I got an image here. This is what the leaf looks like macroscopically without any help of magnification. And you zoom in a little bit more, and you can see these little hair-like outgrowths. They occur frequently on these shoots, the stems. They can occur on the leaves, and even some of the reproductive organs that we see in the plants. Now, some are going to be glandular, and they secrete substances that deter herbivory. And I'll show you an example here on the next slide. But what are some of the functions here? Well, one function is that these trichomes may border the stoma or the stomata, and that can help reduce water evaporation. If you can trap water vapor there, then it's harder for more water vapor to evaporate. Some other trichomes may protect against ultraviolet radiation, protecting the cells that are beneath there from the damages of UV, and then some may help buffer against temperature fluctuations as well. So perhaps they serve as a radiator of sorts. Here's a scanning color-enhanced electron micrograph of one that's glandular, and you can see the structures there on the top probably are going to secrete stuff. And the caption here talks about this being the surface of a tomato plant, and the trichomes do secrete a material that does prevent herbivory because the substances can actually glue or are sticky, and it causes insects to get stuck on there, and that would prevent those insects from robbing the plant of material. Some insects like to pierce into leaves and steal some of the fluids from them. So the pattern of these trichomes is regulated genetically, and they do, and oftentimes they in order for them to understand how genes work, they got to look at what happens when the genes are not working well to understand exactly what role these genes play. So the pattern of the trichomes, you can see here in a normal whatever species of plant this is, the leaf there, you can see the trichome arrangement. And here's a mutant here in which we don't have the normal pattern of those trichomes. And it turns out that you have a mutant that's not able to initiate the growth pattern on there. And so they're looking at the case here when that genetic control is not normal because of a mutation. Then you're not going to get that pattern of growth formation. So you're going to need some proteins that include one called a GL3. And in that case, when you have that gene present, which likely involves in production of enzymes and so on, you get the production of the trichomes on the surface. And another set of genes would provide some enzymes that would inhibit growth in a neighboring cell. But if you don't have, because you don't want them on every cell throughout the entire surface. But if you do have a mutation in one of the genes, then the overall control is lost, and you get a non-normal pattern. Here are some root hairs. This is a radish seedling, and a radish is one of the ones that I suggest that you might use. You can pick up a bag in a garden center somewhere for your plant growth experiment. They grow rapidly, and so it's easy to see or test what kind of factors might affect the growth. Now you're going to pick one for the experiment, one factor was described for you in that experiment here, but these little root hairs actually represent long, thin extensions of an individual cell epidermal cells. So the root hair is a special kind of epidermal cell. And they shouldn't be confused with lateral roots. These are not lateral roots. These things are really tiny and really thin. And they do function to help increase surface area so that the young root that's been forming, the meristem is down here, can do adequate absorption. They end up getting lost as the root ages in this area. The root continues to grow this way, and the parts above it get older and go through more development. In that case, how do the plants continue to absorb efficiently if they lose the root hairs? In this case, many of the land plants, as was mentioned earlier, end up developing symbiotic relationships with mycorrhizae, which are fungi that would help in that absorption for the plant. Now we're going to look at ground tissue. So that was dermal tissue, a brief overview of it. And then for the ground tissue, you think of this as the one that's kind of going to fill in the stuff and would have multiple roles. So there's 3 general kinds. And the lines kind of get blurred between some of these types of tissues and vascular tissue. So we got to be careful with that. But let's just look at them and view them separately for now. We have parenchyma, collenchyma, and sclerenchyma. Okay, and the parenchyma and the collenchyma are going to be living. They're going to have living protoplasm in there, and the sclerenchyma, when it matures, the cells that make up that tissue are going to be dead overall. And you kind of see this pattern here. I found this diagram here as a parenchyma. The cell wall is going to be just a primary cell wall. The collenchyma, the cell wall is going to be a bit thicker, may or may not have secondary cell wall, and then the sclerenchyma with very thick cell walls for the purpose, one of the main purposes for support and protection. Collenchyma will have a similar one. Parenchyma is going to be involved in storage, so we probably talked about that as general for ground tissues. Some may be involved in photosynthesis and some in secretion. The one involved in photosynthesis, for example, may be called chlorenchyma, which is just a specific category of parenchyma. So looking at the parenchyma, and I use what we might call collenchyma as an example here. I mentioned earlier the middle of the leaf in this diagram, and then we have a micrograph showing the cell types that you find in there. You can see they have a lot of chloroplasts in there. This is the case where this is a parenchyma, but it's specialized for photosynthesis. Here, the key thing to all parenchyma, including something like this, which would be chlorenchyma, a specific kind of parenchyma, is that the cell is living. When we say living protoplasm, we're talking about the plasma membrane and everything inside of it is living. And they do provide important functions for the plant, like storage, photosynthesis, and again, secretion. They're only going to have a primary cell wall, and in many cases, these are sometimes the least specialized of your plant cells. Others go down a very narrow path and have just like function like just protection, and that's it. Okay. So that's the same image here. And you can see abundant chloroplasts in this kind of parenchyma. So there's different types of parenchyma. And then the collenchyma. Right away, you can notice here that the materials in the cell wall do pick up a stain when they stain the tissue preparations. This is cross-section through where cells would be, and what's stained red would be that would be the cell wall. You can see that there's actually different kinds of collenchyma based on just the way the cell wall looks. There are sometimes the cell wall looks like it has corners in it. Sometimes it just looks thicker. And this collenchyma, again, the protoplast, the cell is living. Okay. They may or may not, here it just indicates lack secondary cell wall. Secondary cell wall would be more cell wall added to the inside of the primary cell wall. But this one, a primary function would be to support plant organs like support for leaves and for stems. And it's going to allow them to be flexible without breaking. So that's the basic function of collenchyma. And then we have sclerenchyma, and this one here, no living protoplasm, lack living protoplasm. So here the cell's dead at maturity, and the key thing here is that they're going to have a secondary cell wall. They're going to have, and the cell wall is thicker, so it's secondary. And there's going to be 2 general types and both are strengthening tissues. They include fibers and sclerids. And the image your textbook provides for you is that of sclerids, which is down here. So I went and I wanted to share another one. This is a cross-section through a stem of a eudicote, which is a kind of flowering plant in which the seedling has 2 cotyledons, which is likely the seeds you're going to be experimenting with. If you use bean or radish, those are eudicotes or dicot for short. There's actually a technical difference between eudicote and dicot. But over here, there's a key here on the side, which you'll have access to this PowerPoint, so you can always take it and look at it. But one of the places we have sclerenchyma is identified as letter E here, that darkly stained area right there just outside, and on the other side you'll have vascular tissue, and then this is ground tissue in the middle here, which is probably going to be storing things like carbohydrates, stuff like that. And these areas have names. Like the middle of the stem is called a pith, but it has ground tissue in there, right? So, but here's a case where you have some sclerenchyma just to the outside of the vascular tissue. So that sclerenchyma overall is going to provide support for a long stem. So remember, this is a cross section through a essentially roughly cylindrical stem, right? It's a cylinder in shape. The kind of sclerenchyma that you find in this part of the plant would be more of a fiber kind with longer fibers running throughout and strands of the cells that produced that structure, right? The cells built these cell walls. When it comes to sclerids, these are going to be variable in shape. Sometimes we refer to them as stone cells. And let me show you where the living cell would have been that built this. So when we look in here and we go in there, right where I stopped that red line, that's where the cell would have been. So the cell would have been there, there, and there. Those would have been 3 individual living cells that ultimately produce the cell wall that's there. And so the cell wall is stained in red. So I'm kind of outlining the cell wall of one. And these sclerids are going to be arranged in a way that actually provides structural support for other cells. The green outline would be actually the border of another kind of cell that would be living in this case. So this might be some kind of a parenchyma cell, like maybe whatever that function is for it. But you can see that that living cell actually goes in between those sclerids that were produced. So this cell produced this real tough sclerid structure there to provide structural support in there. And here's where some of the lines blur. The sclerenchyma is non-living. And sometimes when you read about these, some of the vascular tissue can also be considered sclerenchyma type tissues. So sclerenchyma, parenchyma, and collenchyma seem to be more broad types of tissues. And some of the vascular tissue could be considered and lumped in there with as sclerenchyma type. For example, xylem, which is non-living, when it has its ultimate function, which is to transport water and nutrients up the plant, or water and materials. So there's a bigger picture of that sclerid there, and again, the actual cell, when it was alive, the cells die when the structure matures, and then you can see this would be where parenchyma cells would occur within there. So those cell walls provide some structural support and protection there. Then we have vascular tissue. There's 2 main kinds. There's the xylem. And the thing to remember about xylem is it's going to conduct water up the plant from the roots to the shoots. And oftentimes that water is taking things that came from the soil, compounds that contain nitrogen, minerals, and water. And then the phloem, which is going to conduct material from the leaves back to all other parts of the plant, including back down to the roots. So this vascular tissue is basically continuous from the roots all the way up to the shoots. It just branches outward overall. So when I show 2 different kinds of stems, this is a dicot or eudicote stem, and this one here on the right is a monocot stem. And you can see that the monocot stem has this characteristic scattered arrangement of these vascular bundles. And those bundles of vascular tissue basically are built with cells that run up and down the stems. They can have both xylem and phloem. And then earlier I was mentioning this looks like that other stem I showed earlier. Outside you'd have your sclerids, but over here you would have both xylem and phloem in there. Xylem going up the plant, phloem going back the other way. The phloem is going to conduct water along with organic compounds like carbohydrates produced by photosynthesis, or hormones or amino acids or other kinds of materials to all parts of the plant. For the xylem, it's from the ground up. So for when it comes to xylem, this xylem is going to be made of a couple of special cell types, and then some others get involved that are in there. But two major cell types, these are going to be dead when this tissue is fully functional, transporting. It's just the cell walls that remain. And you're going to have tracheids, which gives vascular plants their name. Remember, we talked about once you get to vascular plants, they're generally called tracheophytes. Which means plants with tracheids. So they have this kind of cells that are dead. These cells are tapered. So if you look at it and we outline one of the cells, this would be the outline of one of the individual cells, and then another one, and then it tapers down here. And then they cut away the inside there. When you look in there, you're going to see that the individual plates were separated by a plate, but that plate has these pores, sort of pore-like structures that allow materials to run from one cell to the next. So they end up making this what essentially would be a long, continuous tube-like network, very microscopic. And then the more advanced plants, the flowering plants, are going to have these vessels, and those vessels, so the anthophytes. And then there was that one genus, Welwitschia, that we studied, that also has some vessel-like components to it. As well. That was studied in the laboratory, and in the prior chapter was mentioned in lecture. And here the diameter of the actual cell is much wider, and that allows for more efficient movement here, which is one of the reasons that may have given anthophytes, the flowering plants, a bigger advantage over the gymnosperms, because the gymnosperms only have tracheids. So when we look over here at this scanning electron micrograph, which shows cross-section up here on top through xylem, and then going along down this way, you're going to see this is long section, so it's more like we section through in lengthwise there. But when you look here, you have both vessels and tracheids, and that tells me that this is probably a flowering plant and not a conifer like a gymnosperm, because the gymnosperm would not have these vessels here. The vessels are also going to be separated by a wall that would have these sort of like sieves or vents or pores that allow the movement of water to go up the plant. Now, all that's left is the remains of what the plant were, which is the cell wall, to build this type of tissue. There you can see a little closer here, you can see the little pore-like structures there in the plates that separate them, and then same here. These perforations just mean openings in there. And then here you see the electron micrograph on the bottom picture showing a picture of those perforations going through there. Then you have the phloem, and the phloem is actually alive, the tissue, the cells there, and they're going to be generally, these tissues were simplifying them. There may be more types of cells involved here, but some of the main components are just mentioned here for the sake of the survey course. But you're talking about the phloem here. These cells are going to be conducting water and organic materials, hormones, amino acids, and they're going to actually have to use a bit of energy to do this process here. And it has to do with where we're getting the water and these nutrients to be moving. So one of these types is going to be called sieve cells or sieve tube members, depends on what group of plants you're looking at, right? So most vascular plants and the gymnosperms, they're called sieve cells, and there's slight modification when it comes to the flowering plants, the angiosperms. They're called sieve tube members. But in any case, they both have individual cells that have cell wall and living cytoplasm. But separating one cell to the next, they're going to have these plates as you go from one cell to the next here. And the plates have these pores in them, and they act like a sieve. Like a sieve is going to prevent just anything from moving there. We want to keep the cytoplasm in one cell or the next. Okay, so this would be your sieve tube member for an angiosperm, and then that would be the same thing right here. So those would be the same. So in here would be your sieve tube members, and then you're also going to have a companion cell. So this companion cell right here bordering it, you can see the nucleus in one, and then they're trying to point to one, which would be, or at least a group of them right here. And the main thing about these guys here is that the companion cell is going to use energy to pump things like sugars that have been picked up from the leaf as produced by photosynthesis, and that's going to require some energy. So as we pump that into the sieve tube element, anytime you put more dissolved particles on one side of a membrane than another, anytime you have more particles, it's going to create an osmotic pressure. And osmotic pressure drives water from one place to another. So that's going to also cause water to build up in the cell, and that builds up pressure in the cell. And anytime you build up pressure in the cell, and let's say the pressure is higher up here than over here, lower pressure, water or fluids always move from higher to lower pressure. So the phloem requires an active energy input to build up a pressure to create a pressure gradient to move the fluids along here. That's not required for xylem. Xylem is dead tissue, and water is basically just using some basic physical properties of both water and the cellulose that makes up the xylem. So in section 3 of chapter 35, we're going to be looking at the structure of roots, which are going to anchor and help in the absorption of water and materials from the soil. Now, your learning outcomes are to be able to describe key changes that occur in the 4 regions of a typical root. So in other words, you should be able to describe, knowing about the regions, the growth and development that occurs there, which involves cell division, differentiation, and development. And then you're going to be able to explain the function of root hairs, and then describe functions of modified roots. There's different kinds of roots that we're going to look at there at the end. Now, we're going to look at the 4 major regions that are recognized. There's the root cap that protects the root apical meristem, which is down here on the bottom. There's the root cap in the diagram there. And then there's 4 general zones, or 3 zones that are they're not clearly defined. Here's an exact boundary to the next one. They just kind of transition from one to the next. So going in order, the first zone from the tip of the root would be the zone of cell division. Then the cells elongate and then they mature. That means they differentiate and develop into their fully functional cell there. So you can see here, we start at the root cap, then you have your zone of cell division, then elongation, and then maturation. And one way to recognize, just real quick, is you get to the zone of maturation when the epidermal cells mature, and they mature when they form those root hairs that we mentioned in the prior section. So on the left here, you see an actual picture of a root stained. It doesn't show the root hairs, so they may have either been knocked off during the preparation or they didn't go high enough to get those structures in there, or they just missed it when they sectioned through and didn't get any root hairs. So the basic idea is this. If you talk about the apical meristem, which is right here, those cells are going to divide. Some cells are going to produce the cells of the root cap. Others are going to produce the cells that eventually become the part of the root. And as these cells continue to divide, the root moves this way, is growing in that direction, downward. And that direction is by special cells. Then the cells that remain up here are going to start going through further division. They can still divide. And as they divide, they start going down paths of development. Some will become dermal cells. Some will become ground tissue. Some will become vascular tissue. And all of this is regulated by genes and communications within the cells. And then eventually they stop dividing and they're going to elongate, get longer. So the cell grows in length. They grow a little in width, but more in length than width. And then ultimately the cells go through their final differentiation, and that occurs at the zone of maturation. And there those cells take on their final mature functional roles, whether it's conducting water up the root or protecting the outside of the root, being epidermal. So let's take a look here first at the root cap. And we don't have a similar structure in the shoots. Although the shoots are surrounded by leaf primordia, the root tip does require special protection because the root is trying to dig through or burrow through soil. So the sensitive meristem there needs that protection. So there's going to be 2 kinds of cells that are produced when meristem cells grow and then move downward there. There's going to be the columella cells, which are the inner ones, and those are shaded in brown here. Okay, and those are going to function in the perception of gravity. And then you're going to have the root cap cells. And those root cap cells, they're the ones on the outside here. Those root cap cells are built to shed like the way our skin sheds. So as the root grows through there, some of the skin cells shed. And then they also produce mucilaginous, kind of sounds like mucus, right? Fluid that helps lubricate the process. So that's function of the root cap. And what that does is essentially is going to protect those meristem cells that are located just right above the root cap. And then you have the zone of cell division here. And again, once the cells have divided, the root is growing this way. The meristem cells that are divided, just leaving cells behind just above where the meristem is, those cells stay behind and are going to go through further division. They're going to divide and they're going to start to slowly differentiate going down paths. So some of those cells are going to start to differentiate into your early dermal tissue. Remember, we call that protoderm in section 1. Early vascular tissue called procambium. We talked about that also in section 1. And then ground meristem, which is going to be the pre-ground tissue as well. So that's where, if we were to section through there, we would see those tissues there. And that's what this section would actually show, the more mature tissues. You can see the root hairs there. When you get to this point up here, you're already at maturation. These tissues here have already gone through a further differentiation to their final mature state. Oops. So then we go, we're still in the zone of cell division, and there is some control over what those cells do. For example, whether or not you're going to develop a root hair on the dermal cells. That's going to be controlled by a gene called the WER gene, or werewolf, and then one called the SCR, and then they just nickname it the scarecrow gene. The one for the WER gene, or the nickname werewolf, is it's involved in suppressing root hair development. So you don't want every epidermal cell at that part of the root to have a root hair. So you have to have some control. And then the scarecrow gene is involved in whether or not cells are going to form ground type cells or endodermal. And we'll talk about where the endoderm is. Endo is going to be within. It's going to be a protective layer of cells on the inside of the root, as opposed to the epiderm or epidermis on the outside. I'll show you that structure in a bit. But here, in this case here, with the wild type would be the normal gene, that it's not mutated and is functioning okay here. So you can see that if you have the werewolf gene or the WER gene is involved in suppressing. When that gene is expressed, it tells the cell don't grow a root hair. And whether or not it's suppressed or not has to do with these cells communicating with each other. For example, if this cell, again, the gene is normal. If this cell is growing in a root hair, then it's going to be signaling to the neighboring cell, I got it. Don't worry about it. Don't spend any energy growing that structure. Okay. Now, what happens then if you have a non-functional werewolf gene, which is involved in suppressing or stopping that growth, is needed to do that, then every cell would grow a root hair. So again, how do they figure out how these work? A lot of times they work with plants that have a mutation in a gene. So when a gene's not working correctly, you figure out what it does by comparing to that works correctly. Here they're just showing an image of a normal plant that has a normal werewolf gene, and they're using techniques where they tag the proteins associated with werewolf gene, and then you can see it in the image, or they tag it with molecules that help radiate energy that can be detected here. So whenever they glow in the image, you know that gene is being expressed. And then here's the scarecrow gene, and so let's say we start down here at the bottom of the root where we're in the cell division. They're dividing and trying to differentiate. And toward the outside, let's say if we do a cross section of this root here, you're going to have a central area in the root where there's vascular tissue. Then the epidermis is out here, and then this is ground tissue right here. So the ground tissue is there in the middle, and there's going to be a layer of cells just between the vascular tissue in here and your ground tissue. That layer of cells is called endoderm. and the endoderm is made of endodermal cells. So there has to be control as to what those cells become. So as these cells are dividing, they're communicating with each other, and they're telling each other, hey, you're going to be endoderm and you're going to be a ground tissue. And that's regulated by genes. In this case, the scarecrow gene is the wild, in the case of the correctly working one, the wild type, it works correctly and we get the correct differentiation. The inner cell on the inside becomes endodermal, and the one toward the outside is going to become ground tissue, right? And so this is that ground meristem. So it hasn't differentiated, but it's going to divide. And when it divides, the one that moves in becomes endodermal. The one that goes inward and becomes part of that layer, the one that's outside, is the ground. Now, when we have a mutated gene, okay, the proper differentiation doesn't occur. And all you're going to get is ground cells from there. So again, how do you know what these genes do? Well, one way is to figure out when a gene goes bad, how do you figure out and compare it to when the gene is working properly? And there again, they're using those techniques that tag certain protein or molecules associated with that gene. And in this case, here you have the normal expression of the scarecrow gene. And then we go to this zone of elongation, which is the next zone right here. And there, the cells are no longer dividing, but instead they're going to be growing in length. They grow in a little bit of width, but only slightly so. So the cell tends to get longer and gets that classic sort of rectangular brick-shaped type there. So at this point here, again, the cells are no longer dividing. And once the cells finish elongating somewhere around this region, that part of the root is going to stay stuck in that level underground. It's not going to move anymore. It won't move up. It won't move down. That's where it stays for the rest of the plant's life. The only thing that's going to be moving is that tip continuing to move through the soil further. And then we have the zone of maturation, and that's that top zone up here. And this is where you'd be able to recognize it when you see root hairs on the outside, but there's other things to see on the inside. All of these cells and tissues now have taken on their mature function. They fully differentiated and are doing their thing overall. So here, in this case here, the surface cells, the epidermal cells, you're going to see the root hairs are going to be present. You also have some cells that won't have root hairs. The cuticle is still relatively thin. Those root hairs are going to help with absorption here in this young portion of the root. And again, the root is continuing to move further downward. Parenchyma cells are going to be produced in the cortex. And the cortex, when I drew this earlier, you had your vascular tissue in the middle, then on the outside, you have your epidermal cells where root hairs would be. The cortex is this region here between the epidermis and the central vascular cylinder that's in there. That's going to be filled with parenchyma cells, which will do things like store carbohydrates. Roots can store the products of photosynthesis. Okay. Now, when we move and transition from the cortex and getting into where the vascular tissue is, there's going to be a couple of important layers of cells. The first one is colored in purple in this diagram here, and then the next one is right after it in orange. And then this red and blue cell structures, those are the xylem and the phloem, which we discussed in the prior section on tissues. The xylem conducts water up and the phloem back down. But this first layer here that's colored purple, those cells make up what's called the endoderm, okay, or the endodermis. And they form what's called a Casparian strip, okay. And the word Casparian is kind of cut out there. It just says Parian, but it's Casparian overall. And the thing about this is those cells are going to secrete a waterproof material in between the cells called suberin. I think it's suberin. But it's going to be like a waxy type material. And so that means water cannot just sneak through between individual cells. The water and whatever's coming in with the water from the soil has to go through the cell itself. So that provides a little bit of control over what gets into the vascular tissue inside. So that's the function of the endodermis and the Casparian strips that of waterproofing between there. And then the next one, which is color-coded in orange, is the pericycle. And the significant thing about the pericycle is that it sort of serves as a meristem, and from there these cells can grow outward. So again, it's a layer of cells from in here. Those cells can begin to grow outward and form what's called a lateral roots, branchings of the root. And so that means the root grows from within. It's not the same as root hairs. We don't want to confuse that. And those lateral roots will have the same internal structure as the main root itself. It's going to have the central vascular area, the cortex, and the epidermis on the outside. So what is the pericycle? This is going to be the origin of lateral root growth. This is covered both here in the lecture and would be part of the lab that's covered on plant anatomy. And by the way, this structure here, starting with that pericycle, and the vascular tissue within is sometimes called the stele or the stele, or also called the vascular cylinder. And here they're doing side-by-side comparison of a monocot and a eudicote root. They're kind of like zooming in here into this area here. This red stained area is the suberin that's been stained, so you can see where that's where the endodermis is, and a layer of cells just inside would be the pericycle. Okay, this is showing from the other side, so it'd be like they took a section from out here and then they made or drew a model of what we're looking at here, with your vascular tissue on the inside. The red area is where the Casparian strip is, and then to the outside here is your cortex out here. There's going to be some differences in the way the vascular cylinder, the stele, looks on the inside, and that would allow you to identify monocot, eudicote, something that would be discussed a little bit more in your laboratory as you go through it. So make sure in laboratory, you focus on how to tell the difference between a eudicote and a dicote. One of the main ways I tell is that the eudicote is going to have a xylem that sort of makes this star-like pattern. That's the xylem there. You don't see that in a monocot. The xylem is actually going to be arranged in this sort of circular pattern right around here like that. So is the phloem. So more like a circular pattern. And then on the inside, we're going to be filled with ground tissue. And generally the inside, when it's packed with just ground tissue there, they call that generally the pith, which is spelled P-I-T-H. So the pith may function in storage. This is just an enlarged showing the Casparian strip, which is that waterproofing. The cells there make up the endodermis. So that's an endodermal cell. Then this is the pericycle, the origin of lateral roots. You're out over here. This is that circular region where your vascular tissue is. It's sometimes called the vascular cylinder. Why did the presentation do that? Vascular cylinder or the stele. And then you have your cortical cells or cortex cells on the outside. It's great we showed that. Now this shows development. There's these patterns, and the patterns are going to be the same whether you're shoots or roots. This is for the root development and the shoot development. We're going to start with a zygote, like we talked about in the plant life cycles earlier. So we took an angiosperm. Remember, the pollen is carrying two sperm with it, with the pollen tube. One sperm is going to fertilize the egg and becomes the zygote. And then the zygote will go through some divisions and differentiations to form an embryo that would stay dormant until it's activated with water and oxygen and begins to germinate. Germination is just the activation of that metabolism. As it begins to grow and develop, we're going to do that growth and development. The embryo has both already, in its suspended state, has apical meristem cells on both the shoot and the root. We saw that in section 1, and then these are the paths that those cells are going to take to form certain structures and tissues, right? So we're looking at the root. We'll follow down here. You're going to see the same pattern on both with some only slight difference, so it would be worthy of comparison here. So for that root apical meristem here, it's going to cause, one main thing is it's going to cause growth and elongation of the root based on what we saw there. You're going to have your meristem divide, and it's going to produce cells that are going to be in the zone of cell division. Then you get the zone of elongation, then maturation. So when we start talking about maturation, we're going to start developing certain kinds of tissues. Okay. Remember that pericycle is going to be responsible for growing the lateral roots, and it would have its own meristem as well. It'll have its own meristem. And as far as the meristem cells go, remember some are going to be producing vascular tissue, right? They're going to produce procambium as those cells are differentiating. And then we learned we'll learn that there's going to be, we briefly talked about it in section 1 of this chapter, that there's another kind of meristem called cork cambium that does only work to produce the bark out here. Okay, so when you look at the vascular cambium, that vascular cambium is going to be responsible for producing secondary phloem and secondary xylem. Okay, and the secondary xylem is what we refer to as wood, and the phloem becomes part of what's called the bark, which is towards the outside. So there's the inner bark, and then the cork, which would be the outer bark. When you come up here, you're going to see those same patterns. You have a vascular cambium, cork cambium. You're going to get wood, inner bark, and then the outer bark, which is the cork. Okay. Some structures that end up forming there on the apical meristem would be leaf primordia, which become leaves that are going to have vascular tissue in there, ground tissue, and epidermal tissue as they go through maturation. You're going to have bud primordia, which can become lateral shoots like branches. They may even become flowers if it's a flowering plant. And then, of course, that apical meristem does produce more length in primary growth of the plant. So looking at modified roots here, we have some basic kinds of roots here. There's taproot, which is a main root, and they throw in a carrot, but most places you're going to see this, for example, in the flowering plants. This is going to be typical of eudicots. Monocots include grasses. So if you look at grass, they're going to have fibrous roots. So that's a fibrous root system. There's no main middle root. There's just a bunch of similar thickness or diameter roots all growing from the same point here. For a taproot, you're going to have one main root, and then you'll have lateral roots. These are not the same as root hairs. These start from the pericycle deep within the root and grow outward, and they'll grow lateral roots. So if you went out into a lawn or a yard and there was a weed growing and it had these broad leaves on it, and you pulled it up, it's probably going to have a taproot. But if you pulled up some grass, you're going to see fibrous roots there. There's going to be other kinds of roots, and one that's also common is called adventitious roots. And the adventitious roots are going to arise from other structures, like from a stem. So when a root structure that behaves as a root helps anchor and provide absorption of water and things, they're going to be called adventitious roots. An example here, we'll say it would be like this vine. If this vine is growing along the ground, instead of climbing, it grows along the ground. It may grow roots from the stem. And like you see right here, and those roots would go into the ground. And so those will be referred to as adventitious roots. They're growing from a structure other than the root itself. In this case, in my example here of this vine here, that would be growing from a stem, so that's adventitious. There's going to be other kinds of roots. Here's a short list of some of the different kinds. They include prop roots, which function to help hold the plant up. Aerial roots, which plants, they're just living in the air. They're maybe growing on another plant. They're clinging to that plant, and they get all they need, including water and nitrogen, from the atmosphere. And then if you've heard of pneumonia, that's the disease of the lungs, but pneumos means air, and phor comes from phoris. So these are going to allow us to carry air, and these pneumatophores would be like for plants that are growing underwater where there's no oxygen. They'll send up these roots above the water to allow oxygen to go through. I have some images of these. We'll show them here. There's also contractile roots, which can actually move the plant deeper into the soil. There's parasitic roots, which will penetrate a host plant. An example of a plant parasite is the mistletoe. And then there's others that don't even have any chloroplasts. They have to actually steal things from another plant to get it. It's weird relationship, but those things exist. There's going to be storage roots. So some may store food, some will store water, and then there's buttress roots. And the buttress gets its name because buttress is like a term in architecture. If you have a wall and you put a buttress here to provide support for that wall to keep it from, say, sliding outward and then the entire building collapsing down on you. So let's take a look at some of these. Here's prop roots. This is a prop root on a corn plant. The plants that grow corn, they're actually in the grass family, but you can see those prop roots there. And those are going to help hold the plant upright because the corn plant gets pretty tall. Then we have aerial roots, and these are typical of aerophytes or epiphytic. They live on the surface. Here's an orchid growing here, and you can see these roots helping to not only just cling to the surface of some other kind of plant. There you can see the trunk of it, but those types of roots are going to help pull the moisture from the atmosphere, even nitrogen and so on, and as opposed to the soil. So these would be aerial roots. Then we have pneumatophores here. You can see the swamp water in the image below there. There's the water there. And so these pneumatophores reach up into the air and they allow air to get down under the water and in mud where there's little oxygen. So that helps oxygen get down to the living cells under that need oxygen for cell respiration. And so those are living cells and they need their mitochondria need oxygen to make ATP. And then here's a water storage root. And according to the caption here, this particular one that they split open, that root they dug up is from a plant that's in a member of the pumpkin family called Cucurbitaceae. That's fancy. That's a family name for the group. Don't memorize that, just as an example. And in this case, this particular root weighs up to 50 kilograms, which is over 100 pounds. The kilogram is 2.2 pounds. And then here's your buttress roots. This is a fig tree. And these buttress roots are going to help provide some stability here. And this is kind of key in some, especially like in tropical rainforest, the soil is not very deep. So in cases where those roots can't just really dig down and help support and keep that tree from falling over, then you have these buttress roots to help provide some of that stability. So in this section, we're going to look at stems. And the stems are going to be support for above ground organs. So your learning outcomes here are going to be one, to identify the contributions of an axillary bud to plant form. And then two, differentiate between cross sections of a monocot stem and a udicot stem. That's something you're going to be doing in lab as well. So we can become familiar with it here. You need to know a bit about it here, also in lab. As in the lab, you have to be able to identify not only the stem between the two types of plants, but also structures within there. And then be able to describe three functions of modified stems. So, like roots, the stems are going to contain those three types of tissues: the epidermal tissue or dermal tissue, ground tissue, and vascular tissue. And you're also going to have apical meristems, just like with the roots. The shoot apical meristems, though, are going to initiate stem tissue. And then intermittently, that means every so often, as that shoot is continuing to grow in length, the meristem is also going to be responsible for producing primordia, which are young, immature, developing structures that become leaves, shoots, and even flowers. And so looking on the right here, this is a scanning electron micrograph, and in there is supposed to be the cells that are the meristem cells. And then you can see a leaf primordia, older ones, and you can see younger ones starting to bud off, and they're doing so at these intervals. And we'll learn in a little bit that everywhere these primordia are rising are going to be become what we call nodes along the stem. This is going to be more like what you'll see in the laboratory exercise associated with vegetative structure of plants. And this is a section through the apex of a stem. And so just like up here at the top, that was your shoot apical meristem. This is that shoot apical meristem right there. So those are those cells that are dividing. They're as they divide, they're going to begin to down paths of differentiation. They're going to become the three primary meristems from the original meristem. And so they're going to become that procambium, which is the prevascular tissue, the protoderm, which will become the epidermis, and the ground meristem tissue. And so you see some of those labels here, like this is ground meristem. So this is still a very young, immature part of the stem. And these cells still have to go through differentiation, just like that which was occurring in the root, where you had the elongation and then the maturation stage overall. So you'll have to be able to recognize some of the basic structures that you would see here. Those leaf primordia, those would be right there. These are very young leaves. And then here's older leaf primordia. Primordium is singular. And then right where this is part of the stem, this is part of the older stem, and then so on, you're going to have these little buds, and in there are going to be cells that are capable of dividing, and they're called axillary buds, which is the location right between the stem and where the leaf will be. So it's called an axillary bud, and those can produce branchings, lateral branches, even flowers can grow from them. So looking at those leaf primordia, eventually they become leaves along a stem. And a lot of times the way the leaves are arranged on a stem is characteristic of a particular species of plant. For example, in an ivy, you're going to have only one leaf per node, which I mentioned earlier. And so they'll alternate along the branch. So this is called an alternate arrangement. And the arrangement, sometimes they refer to it as phyllotaxis. And there's that term we saw in a prior chapter, that the root philos means leaf. Sporophyll, mesophyll, megaphyll, microphyll. So there's the use of that root in there. Here the periwinkle is going to demonstrate a different kind of phyllotaxis or leaf arrangement where you have two per node. And they'll usually be right opposite each other along that stem there. And that gives it an arrangement called opposite. So you have alternate in the first one, opposite phyllotaxis, and then when you have three or more leaves coming off of the same node, we're going to refer to that as a whorled, which means to go around type of phyllotaxis. In this case, this one has about 8 leaves, but even if you had 3, they're going to be arranged in sort of a radial symmetry around that node, and that gives it a whorled pattern. Looking at the stem here, we have a summer twig or young branch or stem. And then we have one in winter for a deciduous tree that typically drops its branches. We'll focus here on the summer twig, but I mentioned briefly the winter one. In the laboratory, you should be examining at least photos of these, and then you may go outside and see if you can find, though we're in summer right now or heading towards summer. So it's likely there's leaves on there. But you may find an old dead twig that you can look at, examine if it's fallen off of a tree nearby. But anyway, as I mentioned earlier, the place where leaves arise, they arise as primordia and then develop into the leaves, is going to be called a node. So you have everywhere there's a leaf coming off of the stem here on this twig here is a node. The space between them is called an internode. In fact, these terms were introduced in the very first section, the basic structure here. As far as the leaves that grow off of those nodes, the flattened portion is called the blade, and the stalk that attaches the leaf to the twig is called a petiole. So there's your blade and your petiole. And the area between the leaf and the stem is called the axil. Okay, so and this is where you get the term axillary bud. Right, so there you can see there's an axillary bud right there. Okay. And the axillary bud is made of cells that would go through division and differentiation. So it's like another meristem right there, but it's an axillary meristem. And there you can form branches, leaves can grow from there, and even flowers. So if a flower grows from the axillary region, then it would be called an axillary flower. And then you have a terminal bud, and that terminal bud is going to be covered with terminal scales. You can see these little scale-like structures here. And under there is protected under there would be the apical meristem for the stem itself. When you look, if the leaves fall off, the leaves have this vascular tissue as well. So when they fall off, they leave these leaf scars there at the nodes. And if you were to zoom in and look at your leaf scar more closely, you can see these little vascular bundle scars that are there, and that's where xylem and phloem are running. The xylem and phloem runs up and down the branch, right? The xylem running upwards, and then some of that xylem branches off into these leaves, forming those veins. And then the phloem is also part of those veins, and those veins come back, and the phloem runs back down the other way. So if we now look at a stem itself, and we were wanting to compare the difference between a eudicote and a monocot stem. Here the eudicote is described on the bottom and monocot on the top in our list here, but they're opposite here. So this is the eudicote, or dicot for short, for our course, but there's a technical difference between eudicote and monocot, as I mentioned earlier. So the monocot here, there's going to be a big difference here. First of all, you're going to see that the tissue that's found in the center is it's a ground tissue, but they refer to that area as the pith, so it's made of ground tissue there. And you can see ground tissue here as well within the monocot. But one of the big differences you're going to see is the arrangement of the vascular tissue. You're going to have bundles of vascular tissue that run up and down the stem, and this is in cross section. So what those things I just circled there seem to be scattered throughout the entire stem, and they include the vascular tissue, xylem, and phloem, okay, within there. And here for the eudicote stem, there's going to be more of a ring arrangement for them. So you would have these bundles here of vascular tissue that have xylem and phloem. The xylem is toward the inside and the phloem will be toward the outside. And then we have that sclerenchyma support tissue we mentioned earlier. And that's a major difference. So when you look at the cross-section of a stem, you got to be careful because the root arrangement's a bit different. For the root, the eudicote root has that star-shaped pattern I mentioned in the prior section. And it's found in that vascular cylinder in the center of the root. Whereas the vascular cylinder for the monocot has a kind of a ring arrangement of xylem and phloem. So it seems to be opposite for the eudicote in the stem as far as the ring arrangement. The ring arrangement in the eudicote stem is in a ring pattern, but in the monocot root, that's where you find that ring pattern. And then if you look at these stems in general, you can consider a stem a major organ of the plant. If you look at the ground tissue that's towards the outside, say from the area where your vascular tissue is, there is ground tissue. Like you see, I'm going to point to ground tissue with cells that make up that tissue there, just around the area where the vascular bundles are, filling in that area, both just to the outside and inside. And then, of course, on the outside, you have your epidermis, the outer layer, right? Okay. There's ground tissue there, and that ground tissue is different kinds of ground tissue with different functioning. But overall, that region where that ground tissue is going to be called the cortex. In fact, cortex is the outside of an organ. Your kidneys have the renal cortex, for example. Your brain has the cerebral cortex. So general term, it's the external region of an organ in both the plant and in both animal and in this case the plant. That central area where that ground tissue is called the pith. Okay. When you look at the vascular arrangement, the comparison between the eudicote and the monocot, one of your objectives is to be able to know and distinguish between the two types described that. That ringed pattern that we see in eudicotes is one of the reasons that allows for the growth in thickness, the secondary growth. Okay, so that's what this part is talking about, that vascular tissue arrangement. Okay, so if we look at a stem of the primary plant body for a eudicote, this is the eudicote stem. Okay. There you can see here's your xylem and your phloem. The xylem's on the inside, the phloem's on the outside. Okay. Your pith in the middle right here. the cortex right in this region here. Okay, but you look at that vascular tissue. This is what's left behind when the stem matures after the primary growth occurs from the apical meristem, and then it matures, and there's your mature primary plant body. Now, as the plant gets bigger, it's going to have to grow in thickness to support the weight. And the cells that are between the xylem and the phloem, everywhere there's a bundle, right there, those cells, some of them are going to serve as our vascular cambium. That vascular cambium is a meristem where cell division is going to occur, but the cells that come off of here are going to differentiate only into vascular tissue, into xylem and phloem. So what we have here, colored in a darker blue, and you can see the darker blue in the older stem that's now thicker, and the darker red, that was your primary xylem and phloem from the initial growth right here, when the stem was first growing in length. When the vascular cambium develops and begins to produce xylem and phloem, that xylem and phloem is now referred to as a secondary xylem and phloem and represents that secondary growth that gives you an increase in thickness. So this green line here in this diagram is not green in a real plant or a real tree, a eudicote, right? But that green line, those would be the stems that divide. And those that divide and move inward are going to produce the lighter blue colored secondary xylem. Those that move outward are going to produce the lighter colored red or pinkish colored secondary phloem. So basically, the primary xylem stays toward the inside of the stem as new xylem is being added, and the secondary phloem gets pushed outward as new phloem, which is the secondary phloem, gets added to the inside of that. So that's how the growth occurs here in secondary growth and thickness. And remember, only eudicotes are capable of doing this, right? So when we look at a cross-section of an older, say a stem, a tree trunk, we can see that the lighter blue, that was your secondary xylem here. Remember, that's the stuff that transports upward. The light pink color is the secondary phloem, and the green line in between represents the vascular cambium. Okay, and then you can see that every year you're going to get these annual rings, which can allow you to interpret it as the age of that stem or branch or trunk. So those are the familiar annual rings you've seen before. Now, as this stem gets larger, the epidermis has split, and the epidermis needs to be complete, okay? And it's no longer an epidermis. It's going to be transformed into the structure called the periderm. And the periderm for most species typically splits, so you get this split-looking pattern on the outside, giving you that familiar bark appearance. This bark is going to include, so all of this from where the vascular cambium doesn't include the vascular cambium, all of the phloem and anything outside of that is going to be referred to as the entire bark. Okay, but there's the inner bark, which includes the phloem, and then there's the outer bark that's produced by another meristem colored in purple. That meristem colored in purple is called not the vascular cambium, but the cork cambium. And that cork cambium is going to grow as it divides. Some cells move to the inside, forming a ground tissue, and then to the outside, forming cork tissue or cork cells. And that together is called the periderm. And as this trunk grows bigger and bigger, the periderm continues to split, but the cork cambium continues to fill in that splitted area to keep the inside living cells protected. So you need that protective barrier there on the outside. So that's highlighted in this next image here on these older stumps as they grow larger. That xylem now, we might refer to it as the wood of the tree. So now you have these woody plants like shrubs and trees. On the inside, you have some of the oldest xylem called the heartwood, and then the younger xylem as you move outward. You have your phloem. They colored the vascular cambium in this image as we peeled away the layers. They colored it white and your cork cambium in green. The green was used for the vascular cambium in the prior pictures here, but here's where the important part is about that cork cambium. Okay, that cork cambium is like another meristem for secondary growth, but this one is responsible for producing your outer bark that is protective. It's waterproofed, so water can't evaporate off of the trunk, for example, or the side of the branch, right? And it doesn't allow things to penetrate in, especially things that are dissolved in water. Okay, so again, from that cork cambium, okay, you're going to have these box-like cork cells that move to the outside, which are waterproofed. And then you're going to have parenchyma cells that move inward, are going to be referred to as phyloderm cells. And that collectively, as I mentioned a while ago, is the periderm. And here's an image of a section through the outer portion. So let's say that we have a branch, right, like this, or a trunk. And if I just take a little section right off of there, right off of the edge, okay, and then I look at the cross section through there. The outside is here and the inside is here. This would be the outside, and this is the inside right here. Okay, so here's your cork cambium cells right here, right there. Okay. When those cells divide, the ones that come outward form these box-like cork cells. Those that move inward form this parenchyma here called the phyloderm. And all of this together, both the phyloderm and the cork, is called that periderm, which is what replaces the epidermis, or the epiderm. Okay. So there are cells underneath, and if you completely waterproofed your bark, then those cells would not be able to get any oxygen or get rid of their carbon dioxide, because if water can't pass through a structure, then neither can these gases. It's going to be hard for them to penetrate. So in that case, these little openings are left in this bark called lenticels. And so these lenticels are just going to be little areas where you do not have the waterproofing. Okay. And the name of the compound is, it's like a waxy lipidy material, which is lipids don't like water, is unsuberized, because the name of that compound is called suberin, which I mentioned earlier. We were talking about the Casparian strip inside of the root. Okay, so here is you can see the lenticels here on the side of this particular kind of species. Not all species look the same or produce lenticels in the same pattern. Keep that in mind. And then if we took a cross section through this trunk and looked at it, this would be one of those lenticels. This is inside. There's the pith in here. You have some xylem here and a phloem out here. And then you have your outer bark here. And right here, there's that opening. That's a lenticel. This is an area right here where gas exchange can occur. We can take in O2. There's no, so there would be a case where there's no photosynthesis, but those living cells in there still need oxygen for cell respiration, and that produces carbon dioxide, which we got to get rid of. That's the function of those lenticels. Okay. Now, looking at modified stems, there's several kinds. There's a list here, and then there's some images of some of them. There's bulbs, which have fleshy leaves. Think of an onion. An onion is a bulb. Then there's corms, which kind of resemble bulbs, but they don't have fleshy leaf structures in there. And then you have rhizomes, which were mentioned already when we were talking about ferns. As ferns grow as a rhizome, it's an underground stem, and then at nodes, the leaves pop up from there. And then runner and stolon are used interchangeably, and these are an above-ground stem that runs along the surface. And then we have tubers, which basically is what a potato is. A potato is a tuber that we are able to eat. And that's going to be a place on a widened stem that stores carbohydrates, like the potatoes, which are used to make French fries, right? And so these tend to be swollen tips of rhizomes. So the rhizome would be a stem underneath, and then so here it's just an extension of that stem, where you have the starch stored there within there. Then you have tendrils, and tendrils grow off of things like vines, and the tendrils are going to be modified stems used to cling to surfaces so vines can attach to and climb, say, other tree trunk or something like that. And then you have cladophylls, and these are flattened stems that are actually photosynthetic. So on the stem itself, they have cells that have chloroplasts on there, and they can do photosynthesis. This is typical of cactus. The cactus itself, those are not leaves, it's the stem that's photosynthetic, and the leaves have actually become reduced to the spines on a cactus. So here's an example of a bulb. So this is like an onion type there, and you can peel away the leaf-like structures there. So this is that modified stem there, again called a bulb. And then you have a rhizome, and so here this part right here is the rhizome, and the rhizome may be somewhat longer. They just cut off a piece there. And you can see the roots coming off of there. Remember, when roots come off of something other than a root, they're called adventitious roots. In this case, while the rhizome resembles a root, it's not a root. It's a stem. It's just a stem that grows underground. And then here's a node where the leaves grow up above the ground. So you might see stuff like this, for example, in grasses. They'll do these rhizomes underneath. And then runners and stolons used interchangeably. These are going to run along the surface here. So here you have a plant here, and there's the roots here. And then above ground, you're going to have a stem, and then some leaves grow from a node there, and then another node elsewhere. And then if some roots grow down from under here, then those would be called adventitious roots because they're growing from a stem. So again, runner. They're also called stolon. And then we have, this one says stolons and tubers. I'm not sure if that's the correct term, because the underground stems, which this is one right there, would be called a rhizome. So it would be more like a rhizome and the tuber. The tubers grow sort of this underground, right? So this would be underground there. So I'm not sure, based on the usage, stolon is supposed to be above ground, so this may actually be technically rhizome. Okay, so unless those terms in botany are used interchangeably, I'm just not aware of that. So it's maybe an error in editing of this materials that come in your book. Then you have the tendrils, and so you have a vine that's been growing, and then you can see this tendril here, and the tendril is wrapped around the branch of some other species of tree, and that allows the vine to grow upward. And then here's your cladophylls. So keep in mind here, this looks like our familiar prickly pear cactus. Check that out. You can see, I just noticed there's some snails clinging to the cladophyle there. The cladophyle itself, it doesn't look like, it looks like a leaf, but this is the stem. The leaves are these things that become those little, the spines are not thorns, they're called spines, and the spines are reduced leaves. And in this case, the spines have become, the leaves have become so reduced, they don't do the photosynthesis. Instead, the cladophyle does. And then those spines are modified to help protect the fleshy stems from herbivores. This last section is on leaves. And basically, these leaves main function is for photosynthesis. And so your learning outcomes here are to relate leaf structure to its role in photosynthesis. And then to explain how leaf structure affects gas exchange and water loss. So here you're talking about the stomata that are located on the surface of the leaves. Now, the production of a leaf starts off with the primordia. Those come off of the continued cell division, growth, and cell division and development, differentiation from apical meristems. We saw that in the prior section, the leaf primordia growing around the apical meristem. Again, the leaves are the principal site for photosynthesis. In some cases, stems can do that. We saw that with the cactus in the prior example, and then they expand by cell division, and the structure of the plant is determinate. So the leaves typically have a typical size at which they grow to, a range roughly, and then they stop growing. They don't continue to grow the way a stem would. So there are going to be different patterns of leaf structure that allow them to adapt to specific environments, if it's a dry environment or whatever kind of environments you find the plants growing in. So looking at the leaf morphology, there's two basic groups, morphologically speaking, in general, generalized morphology, and they're related to the evolutionary history, which was discussed in the prior chapters. There's the microphylls, which are also called lycophylls, and they appear to be have evolved separate from the megaphylls, which all of the other vascular plants developed. They're called microphylls because they're very small. This was discussed in the prior chapters. And they only have one vein through there and a little fleshy leaf on either side of the vein. And so the leaves are smaller, limited on how big they can get. And they're found in the club mosses, which is the phylum Lycophyta, just to review a taxon, a plant taxon. And so here's a picture of some club mosses here. They don't get very big. The structures, some of the things you went over in the lab, for example, where the spores are produced. This is your sporophyte. Spores are produced there in the strobili, which are it's kind of like a cone, like a conifer. No, it's not a conifer. Then you have your megaphylls. They're also referred to as euphylls, which translates to true leaves. Remember that P-H-Y-L-L stands for leaf. So almost all the other plants are going to have euphylls or megaphylls. So on the image on the right, to help illustrate, you got the blade of your leaf, the petiole, which we mentioned earlier. At the base, you're going to have two structures that grow from there called stipules, and then you're going to have veins within the blade, which is flattened, right? So the leaf basically is specialized for photosynthesis. So if you flatten it down and you face it upward, you have more surface area to do photosynthesis. So the shape of the leaf blade represents a shift, going from radial symmetry, which is just round, to more of a top-bottom symmetry, or a dorsal-ventral. Dorsal would be top, ventral, bottom, overall. So when you have a top and a bottom of the leaf, that means there is an orientation for the leaf. It's the surface of the leaf. The top is actually different from the bottom. And this, of course, is under control of some genes, and they are referenced in your book. And these genes are given names like this: PHB and PHV and KAN. And so here you have a mutant on this side, and then here you have your wild type. And so the leaves just visually look different, but there's another thing that's different. Is the surface, the top of a leaf is going to look different than the bottom. For example, in many of your broadleaf type of plants, you're going to have a lot of your stomata on the underneath part of the leaf. But it turns out when you have a mutation in this KAN gene, there seems to be a problem in the development in that the top and the bottom both end up developing into two tops. So the surface, the top surface and the bottom of the surface are matching, rather than having this the differentiation that's supposed to occur. And again, how do they figure out how these genes act? And what they do is you study mutants and then see and compare it to the normal wild type there. And so I mentioned stipules earlier. Those grow at the base of the petiole. And so here you have the petiole of a leaf, and then there's the base, and this is the stem. That would make that a node. These stipules grow out of there in a pear-like structure. So there's one stipule and there's another. And the interesting thing about these stipules is that some of those become spines. And in our thorny scrub type forest, we have a lot of species that produce these thorns, which are actually stipules. There's one called the cat claw. And it gets its name for a reason. The stipules are shaped like cat's claws, and if you walk near one and you're not wearing any sleeves, they'll stick into your flesh. And I've yanked off a few of them working out in the field before, doing biological work out there, and so it's not fun when that happens. And then the veins. So the veins are basically bundles of vascular tissue. They include the xylem and the phloem. And some other types of tissues that help in support. And you can see here that there is generally a difference between a eudicote flowering plant and a monocot flowering plant, and they do get their names based on the embryonic leaves called cotyledons. The eudicote has 2 cotyledons, and the monocot has 1. But they have some typical characteristics, and one of them is how their veins are arranged within the leaves. The eudicote is going to have more like a network of veins. So this one is the eudicote. And then there's different patterns. So this one is a pinnate pattern, reference to like a feather used for a pin. You have a main central vein and then branchings of veins. Whereas a monocot, the veins are just all running parallel. So that's a major distinction. So when you see parallel leaves, that's a monocot. Okay. That's the same image. And then there are different kinds of leaves. This is mentioned back in section 1. I distinguished between simple leaves and compound leaves. And here you have a compound leaf where you're going to have on the right, so this one's compound. And here you have one main vein in the middle, and then the blade is not entire. Instead, the blade is broken up into smaller blades or leaflets. So this makes this a compound leaf. Now here's your stem, and your stem branches off this way, and this is an individual leaf. So this is a simple leaf. It has lobing on it, but the blade is not completely broken. So the margin of leaves can also be characteristic of a species. Some simple leaves like this will have serrated blades, so the edge would be more like a serrated knife. Okay, some are just going to have an entire, just smooth edge, and so those are all these types of characteristics we can talk about here. So they talk about with simple leaves, you have teeth, which is serrated, indentations, lobes. This one would be more of a lobe type simple leaf. On the epidermis, we talked about the epidermis already, so the trichomes on the surface of the hair-like structures. But we know that underneath the leaf is among the locations where you'll find your stomata, and the stomata are bordered by these special epidermal cells called guard cells. There's a pair there, and when they're going to go under pressure, when a lot of water goes in there, it causes them to bow outward, and that opens the stoma. And that allows for that gas exchange to get carbon dioxide to go in and oxygen to come back out. The leaf does have an epidermis, which includes those guard cells as well. Inside the leaf, you're going to have parenchyma that has a lot of chloroplasts. I mentioned the earlier section, they refer to that specific parenchyma as chlorenchyma. To reflect the fact that it's a tissue with cells that has chlorophyll in it because of the chloroplasts. Remember that just like any other epidermis is going to have a waxy cuticle to prevent water loss, and then you might have those trichomes as I mentioned earlier, and then the guard cells that we just talked about. So if we were to section into a leaf and then look on the inside of it, there's also a difference between monocots and eudicots. And the image in your textbook here and in the lab manual, they only give you a eudicote example here. But you have, and this one looks like it has trichomes right there, and then this one layer of cells right here, that's your epidermis. It's your upper one and your bottom one. Your bottom epidermis would have those specialized guard cells for the stomata. But inside, the cell is right in there. Okay, so you have one layer for your epidermis with a waxy cuticle on top. That's where your parenchyma is, that's photosynthetic, your chlorenchyma. And you can see that it's green, it's got chloroplasts. They even section through a vascular bundle, which is a vein, which has xylem and phloem in there. And then this is an actual micrograph of a light microscope of a thin section through a leaf. And you can see they did use some stain, so these cells are not typically pink there. But you can see that this structure inside, which is called the meso, which means middle, and we know P-H-Y-L-L means leaf, so the mesophyll here is broken up into 2 parts. So mesophyll means in the middle, and here's where you can tell the difference between a monocot and a eudicote. This is a eudicote. Okay. The eudicote has this top layer within the mesophyll that has basically wall-to-wall cells, okay, with very little space between them, and that's called the palisade mesophyll. Okay. And there's that term chlorenchyma. So you're packed there with chlorenchyma. And then the layer, there's a layer within there, lower in the leaf, within the thickness of the leaf, that has these air spaces. So here, these air spaces allow for better exchange. So you'll have the stomata. They have a stoma right there. So when carbon dioxide comes in, it can diffuse around these cells better and even be transported higher up to that palisade mesophyll. And then we can also get CO2 out. So that function of that airspace is to allow for those cells to get and exchange those gases more easily. That area is called the spongy mesophyll, just like a sponge has pockets of just airspace in there. Same idea there. That's how you can tell a eudicote, because a monocot doesn't really have the palisade mesophyll. It's going to look more or less like spongy mesophyll throughout the thing there. So here it mentions how the monocot leaves is not really differentiated into palisade or spongy. It's just going to look the same there. By the way, in here you can recognize and see that there's sections through veins there, which have bundles of vascular tissue. And then there's a layer of cells around there called bundle sheath cells. So there's your bundle sheath. The entire structure would be the vein, and then you have your xylem and your phloem. So then we have some modified leaves to end as we try to end this discussion on the vegetative plant structure. Sometimes you have modified leaves that look like flowers, but they're not, and those are called bracts. And this one seems to be pretty popular in landscaping. I do see it in landscaping around our area here. And if I'm not mistaken, I think this plant is relatively good with drier conditions. We live in a subtropical, semi-arid climate, lots of warm temperatures. Sometimes we have years without a lot of rain. So that would make this one kind of good for landscaping. But you can see here that the actual flowers are growing here, and they're white. But then you have it surrounded by modified leaves that are colorful and have a similar function to petals of a flower to help attract pollinators. Those floral leaves, or leaves modified to look like flower petals, are called bracts. Okay? Or floral leaves is another term. I've always called them bracts. Some will be modified to spines, like the cactus. So remember the cactus, what looks like a leaf is actually the stem, called the cladophyll, and the leaves have become very reduced to form spines. And when you reduce the leaves, that helps you reduce water, which is an adaptation for cactus to live in the desert. And then animals that are starving for both water and food are going to be deterred because those spines help provide protection. There's also leaves associated with reproduction, and then those leaves can grow an entire plant by asexual reproduction. And then succulents, these are going to be plants like cactus, but not usually a cactus. There are other kinds of plants that are succulent, which means they retain a lot of water in the flesh. And they have these window leaves, and these window leaves provide this clear passageway for light to go and reach parts of the plant underground, where you can have photosynthesis occurring there underground. That's pretty interesting. And then you have shade leaves and insectivorous leaves. Now, the image on the right is of the insectivorous or carnivorous plants, and they're modified leaves for capturing and digesting prey. Shade leaves, so let's take a look at that one first. The shade leaves are going to be very, very broad and large. So if you have plants that are growing in a shaded area under trees, say in a dense forest, those plants are going to tend to have much bigger leaves, so there's going to be more surface area to catch whatever light is able to hit that lower level there. So those would be shade leaves. And then the insectivorous leaves, those are made to trap animals like insects. And you're probably familiar with a Venus flytrap, which you see a picture of there on the top right. There's your Venus flytrap. When the insect lands in there, that triggers the leaf to close shut, and inside it has a fluid that helps digest and break down the fly, and then it absorbs those nutrients. And then you have pitcher plants, and a lot of these are pitcher plants, like all across the middle. Those are different species of pitcher plants. And there, when the insect lands in there, they have a hard time clinging to the inside, and they drop down to the bottom where there is a pool of fluid in there that includes those digestive fluids. And then the sundews, those are going to secrete this sticky stuff at the surface that has this digestive properties, and then that traps them and breaks them down. Now, why does this kind of structure and evolve in these types of plants? And one seems to be that there's a connection between where these plants grow and the lack of nitrogen in the soil. So where do you go for nitrogen? Go to an animal. The animal has proteins in there. Amino acids have a lot of nitrogen, so you can break those down and get that nitrogen you need to build your own proteins and DNA. So those are pretty fascinating types of plants.